Erwin Chargaff by Ayla and Bahar. Through careful experimentation, Chargaff discovered two rules that helped lead to the discovery of the double helix structure of DNA. It was known that DNA is composed of nucleotides, each of which contain a nitrogenous base, a 5-carbon sugar, and a phosphate group. The possible 4 bases are adenine, guanine, cytosine, and thymine. Adenine and guanine are purine bases, and cytosine and thymine are pyrimidine bases. However, it was never known how these bases paired up until Chargaff's discoveries. At first, Chargaff noticed that the DNA contained equal amounts of adenine and thymine and equal amounts of cytosine and guanine. These equalities provided clues into the chemical pairings that make up the double helix. In addition, Chargaff also found that the amounts of bases vary by species, an indication that DNA, not protein, might be the genetic material for life. Chargaff became interested in DNA in 1944 after Oswald Avery identified the molecule as the basis of heredity. He saw evidence in support of the belief that living species differed because of their differences in their DNA. At this stage, Chargaff pictured DNA molecules as a Mobius strip, which could split along its center line with each of the two halves inheriting the twisted topology of the parent strip. In 1949, Chargaff discovered that the proportions of bases in DNA depend on the species that the DNA comes from. This was a major break from what scientists had believed until then. Chargaff's first rule holds that in a double-stranded DNA molecule, the amount of guanine equals cytosine and the amount of adenine equals thymine. This strongly hinted towards the base pair makeup of the DNA, although Chargaff was not able to make this connection himself. For this research, Chargaff is credited with disproving the tetranucleotide hypothesis by Phoebus Levine, which said that DNA was composed of a large number of repeats of GACT, meaning it did not have the variation needed to be genetic material. Chargaff, however, said that the number of possible arrangements of bases was a truly enormous amount, and DNA must be the agent of inheritance.
Chargaff's second rule holds that complementary nucleotides are met with almost equal frequencies in single-stranded DNA. Hence, the percentage of adenine is the same as thymine, and the percentage of guanine is the same as cytosine for each DNA strand. He demonstrated that the sum of purines always approximates the sum of pyrimidines, an obvious consequence of the base pairing nature of the DNA double helix. This showed that the composition of DNA varies from one species to another, in particular in the relative amounts of A, G, T, and C bases. Such evidence of molecular diversity, which had been presumed absent from DNA, made DNA a more credible candidate for the genetic material than protein. He said while there were only 4 nucleic acids, as opposed to 20 amino acids, the number of different proportions in which they could exist and their different orders provided a basis of complexity sufficient for the formation of genes. He also realized that there must be as many different types of DNA molecules as there are species. In 2006, it was shown that this rule applies to 4 double-stranded genomes: the eukaryotic bacterial and archaeal chromosomes and the double-stranded viral DNA genomes. It does not apply to the mitochondria and plastids, nor to the single-stranded viral genomes or any type of RNA genome. The experiment. First, he fractionated out nuclei from cells. He then isolated the DNA from the nuclei and broke it down into its constituent nucleic acids. Then, using paper chromatography, he separated the purines and the pyrimidines. This was done on the basis of the solubility of the substances being analyzed. A piece of chromatography paper was dipped into the solution, and the different components of the solution traveled different distances up the paper. Now that he had each of the bases isolated, he then used UV spectrophotometry to calculate their relative amounts. Because each base absorbs light of a different wavelength, he was able to determine how much of each base was present in DNA. Chargaff's conclusions revolutionized the biological sciences. His discovery helped Watson and Crick to determine the structure of DNA through base pairing to propose a double helix structure for DNA. But why is correct base pairing an important discovery? Correct base pairing is essential for the faithful replication of DNA. Base analogs are molecules that replace standard DNA bases during replication and stop DNA polymerase and terminate replication. They're effective antiviral and anti-cancer agents against diseases like hepatitis, herpes, and leukemia. Additionally, Chargaff's rules tell us how much we can read the sequence of nucleotides on one strand of DNA and immediately know the complementary sequence on the other strand. Chargaff's DNA work was never recognized by the Nobel Prize Committee. He became a fierce critic of molecular biology, particularly in techniques like gene manipulation and cloning, which he believed were incredibly dangerous. Thank you so much for watching. We hope that you learned new facts about our scientist, Erwin Chargaff.